Hello friends, welcome to this lecture. Drug discovery. So where does drug discovery all begin? It begins in a lonely lab where an innocent scientist makes a discovery and goes, Eureka, Eureka! From your high school chemistry, you might be familiar with Archimedes, who found out how to differentiate gold from other compounds. So similarly, this innocent scientist will go crazy and run around the lab crying, Eureka, Eureka! So where does this all begin? It begins with target identification and validation. So this is a picture of a clockmaker. So the clockmaker looks into the cogs and wheels of the clock so that he can understand the mechanism. And when something is wrong, he can find out which cog or wheel has gone wrong. Similarly, scientists will look into the disease mechanism and find out which molecule or which pathway is involved in the disease. So I want to bring to your attention the familiar disease, which is depression. You might be familiar with SSRIs. Serotonin reuptake inhibitors are used widely as treatment for depression. The most famous is Prozac, of course, which is prescribed to many hundreds and thousands or millions of people around the world. So in this case, how did they first figure it out? So the scientists studied the disease mechanism and they studied the presynaptic membrane and found out this target SLC6A4. Which is the serotonin reuptake molecule, and then they design compounds which act on this target. So that's target identification. So that's a familiar case for you. So identification is not enough. You need to validate it. You need to show to a high degree of certainty that it is in fact the correct target. So how do we do this? We do this by using cell culture models or animal models that the target is in fact involved in this disease. So that is validation. So after target is identified and validated, you can go on to lead identification finally. So you might be familiar with this game where there's a picture or a pattern of a house or a person or an animal, and then there's a missing link. So in this case, the missing link is the red piece, and when you get the red piece, it completes the puzzle. Similarly, scientists can design compounds which act on the target molecule and regulates its activity. So that is lead identification. So how do we do this? We do this by HTS, high throughput screening, where you have hundreds and thousands of compounds and screen it for activity against the target compound, right? Or else they can study the chemical structure of the target and design using chemistry knowledge, rationally design molecules which bind on the target and regulate its activity. And that's not all. You can identify lead compounds from natural systems, which is plant-based products like what our ancestors used to do in Ayurvedic medicine in India or Sri Lanka, where I'm from. Or else you can get compounds from the bottom of the ocean, because that's a completely untapped territory. And also they get compounds from snake venom, which is used for many blood diseases, right? So that is lead identification. And lead identification is not enough. You need to go into preclinical testing. Because why? Because we did all this lead identification in cell culture models, but as you know, my body is not just one type of cell, it's many types of cells. So we need to test it in a physiological system, but obviously you cannot do experiments on the human body yet, because it's a completely unknown compound, and it might have disastrous consequences for human beings. So we test it out in the next best system, which is animal models. The innocent mouse is used in many cases because of ease of use. Of course, we feel for the animal models, but then we need to do it for humanity. So we do preclinical testing. We do compound pharmacology, where if it's an oral compound, when the animal model is given the compound, how much is released to the blood, how long it lasts in the blood. And you do pharmacological assays where you study the physiological activity of the compound, right? And then you do preclinical toxicology assays. The most important are liver toxicology and kidney toxicology. Because why? Liver and kidney are the front runners in defense against foreign molecules. So they are very prone to toxic effects. So you do those toxicity screens, but that's not all. You do toxicity screen for all organ systems, the nervous system, the blood system, even reproductive system. Reproductive system is specially tested for mutation or where the drug might have effect on the future generations and so forth. So using all these preclinical toxicity assays, we get this important readout, which is called NOAL, which is no observed adverse effect level. So what is NOAL? NOAL is the highest dose at which there's no observable or appreciable toxicity in the animal model. So why is this important? It's important because it is used to derive the proper dose for the first in-human clinical trial. So that's why NOAL is very important. You can look at the graph. It explains, self-explanatory. Hope you really enjoyed this lecture, which is drug discovery, and I really enjoyed explaining it to you. Hope to see you at the next lecture. Ciao for now.